very specific changes on this gene called DCAF4. And so that's what all that kind of means, and kind of the mechanisms behind these mutations on this gene. And so I'm not gonna go into great detail about data and statistics, but of course as Mr. Wright already covered very well, lung and bronchus cancer is essentially the leading cause of death from cancer. In the U.S. in 2015, there were 218,000 new cases and 153,000 deaths. In North Carolina, that number was 8,100 new cases and 5,500 deaths. And so obviously we need to talk about health disparities and social determinants of cancer and lung cancer. So it really is the interplay of several different factors. So when we talk about things like social and environmental factors, so institutional racism is a big one, and can lead to a lower level of health care, lower level of quality of care, and can also lead to lower level of lung cancer screenings as well for folks who are at higher risk for lung cancer. Also, behavioral factors such as higher levels of smoking and drinking and lower levels of physical activity, which obviously those interplay with those environmental factors, because if you don't have access to spaces where you can exercise, or you live in a neighborhood where there are corner stores that sell cigarettes and alcohol, you're more likely to use those substances. And so health disparities and genes or genetics. So when we talk about sort of the intersection here between genes and health disparities, what we're talking about is are there certain types of gene mutations in African American populations in particular that put these folks at a greater risk for lung cancer. Also, we're talking about things like is there something about genetics that can make tumors more aggressive in certain populations. And so we should probably take a little step back here and talk about what DNA deoxyribonucleic acid actually is. So you're probably all familiar with that classic double helix shape. What it basically amounts to, it's pretty simple actually. It's a sugar phosphate backbone, and then you have your four nucleic acid base pairs. You have adenine that pairs with thymine, and guanine that pairs with cytosine. And so this is kind of the very brief version of how we go from DNA to proteins in our cells, which can determine a whole variety of different things about us, including diseases like cancer. So essentially, when cells divide, that double helix shape actually unwinds itself, and it turns into you basically end up with two single strands, which those two strands become the template for new DNA to be made, essentially. And so there's an enzyme. So I won't go too deep into. I won't go too deep into sort of the biochemical and enzymatic processes that are involved, but basically there is an enzyme called DNA polymerase that gets some help from a short strand of DNA, and essentially what happens there is that that short strand of RNA plus that DNA polymerase helps those base pairs grow into a chain of DNA, essentially. And of course, along the way, there could be mistakes that are made, and those cause mutations. And those mutations can end up in all of our cells, essentially. And so there are actually genes that repair those mutations. But sometimes that DNA repair gene doesn't catch all of those mistakes, and so that leads to those mutations or those changes being incorporated into the cell. And so sort of the next step is this molecule known as pre-messenger RNA, essentially. So messenger RNA. That's responsible for bringing those, so essentially bringing the amino acids, which are the building blocks for proteins. It basically takes it out of the nucleus, which is where most of this process takes place, and transfers everything over to sort of the outer part of the cell. And into a complex known as ribosomes, which are basically made of RNA and proteins. And it's at that point, essentially, again, there are sort of a lot of steps in between, but that is essentially at that point where proteins are made, and proteins and the way that they kind of fold and twist and turn can potentially determine a lot about our health. And so I just want to talk a little bit about different types of genetic mutations and kind of what can happen. And so there are four basic ones. You have so insertions. So you see here these sort of all these different letters: the C's, the T's, the G's, the A's. Occasionally, extra base pairs can be inserted into that chain. Sometimes those base pairs can be deleted, leaving you with a shorter chain than would normally occur. Sometimes you get a substitution where one base pair is substituted, like it says, for a different base pair, and then those base pairs no longer align like they normally would. And then you have something called a frame shift, where essentially, again, kind of what it sounds like. It's basically that the base pairs shift in one direction or the other, and so they do not line up as they normally would in a normally functioning DNA molecule. And so, so what are the various different causes of gene mutations? So some of them, like we kind of already discussed, some of them just occur naturally, and sometimes those changes end up in our cells, and which I'll talk about a little bit in a sec. They're not always bad. Most often than not, they are actually pretty innocuous, but sometimes they do lead to diseases. But they can be caused by different behavioral factors like smoking and secondhand smoking, excess drinking, and also nutritional and dietary factors. And some mutations can be caused by certain viruses. And then, of course, you have your environmental factors as well. Prolonged exposure to the sun and the radiation from the sun, radon, and then other chemicals, asbestos, things like that, and other types of toxins can cause these mutations. And so I kind of touched on this already, but so the consequences of these mutations. So lots of times they don't really do much of anything that we know of. So not much changes. They're incorporated to the cell, but are pretty innocuous, and sometimes they are actually beneficial, and they can actually improve things. And then sometimes you have genetic disorders, so Down syndrome, muscular dystrophy, cystic fibrosis, and then of course cancers. And so genes are involved in several facets of cancer and lung cancer specifically. So cell division and sort of the growth of cells as well. Our cells are meant to stop dividing at some point along the line, but that does not always happen. And then when you have this sort of out of control cellular growth, that can lead to tumors. And cell death, of course. So yeah, our cells are programmed to actually die, and so that is a natural part of the cell cycle. But sometimes the genes responsible for that cell death can acquire mutations, and they no longer those cells no longer die like they are supposed to naturally. And then we have, like I mentioned earlier, DNA repair type genes, which hopefully most of the time are there to repair those mutations that can be deleterious, to repair those mutations, but that does not always happen. And then of course it can also play a big role in the growth of tumors and their malignancies as well. And so I want to talk a little bit about our team's previous research that has kind of led to what we're doing now. So a little while back, we actually compared normal lung tissue to tumor tissue in the lung, and we used the method known as genome-wide association studies, or GWAS. And we were looking for very specific variations on those genes called single nucleotide polymorphisms, which basically are just small changes in those base pairs that we talked about earlier. And so when we looked at that, we actually found that the DCAF4 gene, when it had these SNPs or these single nucleotide polymorphisms, having that SNP can potentially put folks at greater risk for lung cancer. And so so what we are doing now in our current research, and you may have heard about this on the news, we are using a technology known as CRISPR, which is essentially it's a gene editing tool, so what that does is that allows you to target specific genes like DCAF4, and actually allows you to interrupt its normal function. And so what we want to know, and so when you interrupt that normal function, you create what's known as a knockout gene, essentially. And so what we want to know is when we create these knockout genes, how does that play a role in things like the growth of cells and cell death and the cell cycle and so on. And so let's talk a little bit about what CRISPR is. So interestingly enough, CRISPR is a naturally occurring sort of technology, I guess, in bacterial immune systems. And so this technology that bacteria have developed, this actually allows them to protect themselves against viruses. And so we have been able to take advantage of that by basically using a sequence of DNA and a short sequence of RNA to be able to target specific genes. And so we also, when we design experiments using CRISPR, we also use what's known as a control vector, and that control vector does not target a specific gene. So in that way, we can compare what the CRISPR-Cas9, which is a protein essentially, we can compare what the CRISPR complex does to what the control vector does. And so what have we done to this point? So we actually created we've created our own CRISPR-Cas9 complex, and what we've done is we've created this DCAF4 knockout gene as well as the control vector. And so I'm not going to cover any of this, but this is basically what that CRISPR model looks like. So just to give you a sense of what that actually would be. But yeah, and there, right there you can see there, that's where the DCAF4 target sequence is. And so this right here, so this is the DCAF4 knockout information, and it's actually incorporated into a lung cancer cell line. And this is a different lung cancer cell line. And so we have ran some tests or assays to see how the DCAF4 knockout gene affected the behavior of our lung cancer cells and cell lines, and found that there was actually a significant decrease in the cell growth rate when we compared it to the control vector. And yeah, so that red line, as you can see, it's not quite as severe. That is the DCAF4 knockout right there, and that one that's shooting way up is the control vector. And this is in the other lung cancer cell line. And so we also looked at the effects of the DCAF4 knockout gene on the growth of these cell lines, and found that the DCAF4 knockout gene had a significantly reduced ability to form colonies of cells, and that's what that looks like. So yes, you got the vector there on the left, the control vector on the left-hand side, and then our DCAF4 knockout gene on the right-hand side. And you can see, and actually the next image is actually a bit more stark. But as you can see, when you compare the control vector to the DCAF4 knockout, you can see for DCAF4 knockout, there is the reduced ability to for cells to grow and for colonies to form. And then we actually treated one of our cancer cell lines with chemotherapy. And we found that the knockout gene actually had significantly higher tumor cell death rates when we compared it to the control. As you can kind of tell here with this graph on the right-hand side. And so this is the chemotherapy exposure, and then control essentially. And then we used some online tools and publicly available data sets, and we actually found that 3 different variants or SNPs, like we discussed earlier, of the DCAF4 gene were positively associated with lung cancer risk. And so further, we essentially we found that these 3 specific SNPs were more common in African populations as opposed to European populations. And so this is just the frequency of those alleles or of those different genes or those SNPs, showing that they were greater in African and African American populations as well. And so what's coming up? So yeah, so we'll basically what we're going to do next is we're actually going to use a colony of mice to basically look at tumor growth delay when we compare a naturally occurring DCAF4 gene compared to the knockout version of the gene. And we're going to also, we partnered with the Duke Bio repository or the biobank to obtain 100 pairs of both archived normal lung tissue and also tumor tissue samples from both African American and Caucasian populations. And we're basically what we're going to do, as we kind of talked about earlier, is we're going to look at how those proteins are created and expressed in the DCAF4 gene. And then we're going to publish the results of all this wonderful, exciting research, and we'll write some grants for future funding. And yeah, so thanks for having me, and thank you very much to the Lung Cancer Initiative and to the V Foundation in support of this research.
So white button. Okay.
Yeah.
There we go. All right. So I am director of the Immune Monitoring Core Lab at LCI, and we've been working over the past year, give or take a bit less, on non-small cell lung cancer, trying to dig further to better understand racial disparities between African American and Caucasian lung cancer, looking at the biology of things, and then we go pretty deep into this. So the title for the talk is Non-Small Cell Lung Cancer, Immunokinetic, Racial Disparities in Host and Tumor Immune Interaction. So let's start first by trying to bring back this discussion to a national picture. What are the cancer stats for this year for lung cancer and other cancers? So you've heard earlier that lung cancer is basically the #1 cause of cancer actually in the US, with an estimated 174,000 people who will actually die this year alone. And in terms of incidence, lung cancer rank at the top actually together with prostate and breast cancer. So we hear a lot about those two cancers. There are some very vocal advocacy groups. Lung cancer less so, and I hope, group like this one and nationwide, we can actually bring this message out there. It affects everyone, always, all ages, younger and younger, and not only smokers. Something important to keep in mind, and it kills a lot of people every year. So when you break down those numbers in ethnic group, you've heard that nationwide, actually, African American men in particular are more likely to be diagnosed with lung cancer than pretty much any other ethnic group, and are more likely to die of this. So in North Carolina, interestingly enough, our numbers are similar between Caucasian male and African American men. Not because African American men are doing that much better, it's because actually Caucasian men are not doing as well as the general population of the US. Maybe due to the fact that historically we are a tobacco producing state, and there has been a lot of people consuming a lot of tobacco back in the time, plus right on map, you can see with the mountain and everything. So we have a lot of environmental factor in North Carolina that may explain why lung cancer in North Carolina is not as good as a prognosis as it may be in other states across all ethnicities. Now, looking at a different metric, and a very important metric actually, nationwide, when patients are diagnosed with lung cancer, how far along are they in their diagnosis? Is it an early disease that can be resected surgically and cured, or is it a later disease that will be a lot harder to treat because metastatic and because requiring basically systemic therapy? And as it turns out, African American patients nationwide are less likely to be diagnosed with an early stage disease and more likely to be diagnosed with a more advanced disease. So that too is part of the disparity map, not only the incidence, but how late in the disease actually patient may be. So we've heard a lot about why do we have those differences between Caucasian, African American, men and women. And there are many, many factors that contribute to this: socioeconomics, culture, diet, stress, environmental factors, and also biology. And those factors do not act in silos. They are really interacting and intertwined with actually one another. So something to keep in mind. So I do emphasize here biology, because I'm a scientist. That's something I can measure. I'm not I don't have an PH or anything like that, so I will leave this to the professional. But I can actually tell you more about biology. And why do we care about biology in lung cancer in particular? One word to that, immunotherapy. So you all have heard about those great advances, revolution in cancer care across all cancer, lung cancer and otherwise, where we are changing something that used to be a terminal diagnosis into a chronic disease. And that's actually a revolution. We can live with lung cancer. We can live with cancer in general, and not for a few months, for years, and keep good quality of life. And that's something to keep in mind. It doesn't work on everyone. We still have a lot of work to do, but this alone has been actually over the past 10 years a huge revolution in our world. In lung cancer in particular, so that's a modern history of immunotherapy. When did it really start, and where are we at now? It started not even 10 years ago. 10 years ago, the first immune checkpoint inhibitor, ipilimumab, was approved by the FDA to treat late-stage melanoma. Since then, I circled in red here every single FDA decision that affects actually non-small cell lung cancer. First, lung cancer was the second cancer that got FDA approval to be treated with immunotherapeutic drug for late stage, for incurable stages, and as of now, it moved to frontline therapy. Most patients will have access very early in their care to immunotherapeutic drug, and this happened this year. No matter what the biology of the disease is, most patients will have access to it. It's expensive, a bit of pushback on the healthcare economics part of things, but that's where we are now with non-small cell lung cancer. And immunotherapy is a very, very peculiar type of drug if you think about it. So when you think about cancer care, you think about those drugs, those devices, those interventions that are targeting the drug, removing targeting the tumor, removing it, killing it in any way, shape, or form. Immunotherapy uses a proxy to do that. Immunotherapy targets the immune system, target our own defense, our own immune defense, to actually eradicate and contain tumor. So here you can understand that when we think about the biology of cancer in the context of immunotherapy, we have to look at the tumor, we have to look at mutations, we have to look at proliferations and this kind of thing. But we also have to look at the immune system as a whole, because in immunotherapy, that's what we are actually targeting, not the cancer. We are targeting the immune system. And how one and the other actually interplay and interact with one another. Which leads me to the research we are currently doing, funded by the V Foundation, the North Carolina Lung Cancer Initiative, and we are doubling those funds with internal funds from the Leon Levin Foundation. So we have overall about $500,000 that we are actually injected into this research to really try to better understand how tumor and immune system interact with one another. You see the word here, dynamic functional. So fancy word, but it means that we are not looking at a steady picture. We'll be looking at patients over time. We will be following patients from diagnosis with a late stage non-small cell lung cancer and all the way through the first 6 months of treatment, which really gives us a very dynamic picture of what's happening when patients present with the cancer, but what the drug is actually doing to interaction between the immune system and the cancer itself. So looking back at our data and the LCI experience, so when we were actually designing this project, it was 2017, we were looking at our data from the previous year, so we had seen at the time 1,300 lung cancer patients. Of those patients, about a third had PD-L1 positive tumor. So what does it even mean? So here I'm going back to the biology of things. How biology is important for immunotherapy. So when in 2013, nivolumab, the first immunotherapeutic drug, was approved for non-small cell lung cancer, it stemmed from a clinical trial headed by Mr. Meyer's Squibb. And what they had found in this clinical trial, we had this drug, nivolumab, immune checkpoint inhibitor, and patients who had this PD-L1, programmed death receptor ligand 1. So it's a very, very ominous name, but it's something, it's a switch tumor can use to just shut down the immune system. As soon as an immune cell gets close to them, they shut it down. And they need PD-L1 to do that. Nivolumab blocks that, prevents that. So if the tumor expresses very ominous pain, those immune checkpoint inhibitors should be working. And that was at the time the reasoning. If you have a tumor with this PD-L1, use immunotherapy. And it became actually a requirement. A pathology lab had to determine that the patient had a PD-L1 positive tumor for the patient to be eligible for immunotherapy. So a third of our patients, give or take, were technically eligible for immunotherapy. 1 in 5 patients at LCI are African American. And when you look at how many of them received immunotherapy, the answer is 20%. We've lost actually 10% of patients that technically should have been eligible to immunotherapy that did not receive the drug. And that's one of the questions we are trying to understand. Is it a biology phenomenon? Is it that African Americans express less PD-L1 or have less immunogenic tumor or less interaction between tumor and immune system? Or is it just an access to drug? When this drug is so expensive, there are at times pushback where patients will be directed toward first chemotherapy, and if it doesn't work, they move back to immunotherapy, or something else that we have not thought of yet. So we have developed at LCI, my lab and a couple of the labs, this Immunokinetic platform where to probe actually interaction between the immune system and cancer. We're actually collecting both. We are looking at tumor biopsies and blood samples. Tumor biopsies, because that's where everything happens, that's where everything we want to trigger actually happens. And blood, because that's the highway of blood cells used to get wherever they are needed. Cancer in this particular case. And we have bring together a team of cell biologists, immunologists, molecular biologists, and pathologists to actually try to reprobe at the cellular and the molecular level the interactions over time between our immune system and the cancer in the context of patients receiving immunotherapy or chemotherapy. So that's our first specific aim for the group. That's what we plan to do and we are currently doing. So we are collecting, we are planning to actually look at tumor and systemic immune system, and we are collecting tissue, tumor biopsies, and blood samples from the same patients across the 6-month period. So not all patients receive the same treatment. Some patients will receive chemotherapeutic drug, some patients will receive immunotherapeutic drug, first line, based on this PD-L1 story. And some patients will start with chemotherapy, hit a roadblock, will start progressing, the treatment's not working, and then be channeled to immunotherapy. So you can see our enrollment target for each arm of the study, and the second arm actually, it doesn't look so nice actually, and it's actually good news. So I was telling you up to March, April this year, patients had to be PD-L1 positive to be eligible to receive immunotherapy first line, chemoimmunotherapy first line. Reanalysis of the CheckMate 0517 clinical trial this year have shown that actually no matter whether a patient express PD-L1 or not in their tumor, everyone benefits from immunotherapy. And we needed those data to change our standard of care, which means that we had to rewrite our protocol because we wrote in our protocol that we would collect patients from the second arm only if they had PD-L1 positive tumor. But most patients that receive that do not have PD-L1 positive tumor anymore. So we had to resubmit amendment and get it. And now most of our patients actually that we'll be seeing in the next few months will be part of the second arm, so we'll be catching up pretty quickly. And that's what we plan to do. A lot of science. So we have developed this immune mobilization and polarization assay, IMP for short. And what this assay does, it actually probes a set of 59 immune variables, looking at 6-cell immune cell lineage, and both in terms of distribution and functionality. So, in short, what we are trying to find out is the immune system doing anything, whether it's in the blood or in the tumor itself? And if it does something, is it does something productive? If it's something where immune cells are fully mobilized, fully activated, and actually doing their job, or they've already opted out and are kind of collapsing somewhere in the body where they are there but are completely not productive. And that's where you see this gradient from cold to hot, trying to determine basically how hot the immune system is. The idea being the immune system is here to contain and eradicate tumor, immunotherapy is aiming really to unleash this phenomenon and really get the immune system go wild after the tumor. So we are expecting very, very hot immune system to really contain and neutralize cancer. So to give you actually an idea of how we can actually use these tools, we already have collected data focusing specifically at racial disparities between Caucasian and African American, and using publicly available data from this 10K Immunome database, we've been able to extract from our 59 variables 14 that were publicly available, and try to understand if there are natural differences between African American and Caucasian individuals that could explain why one group may fare better when exposed to immunotherapeutic drug or when diagnosed with cancer. And what we found is, well, for the most part, actually, we are all the same, which is good news. But when actually you look at very specific immune phenomena, what we can see, for instance, here is that we see something called low terminal differentiation of effector CD8 cells. So does it mean that African Americans somehow are immune deficient? The answer is no, because when we look at other immune effector, B cell, for instance, in this particular cohort, African American subjects tend to have more activated B cells. So what it means is that different ethnic groups as a whole tend to have to allocate immune resources slightly differently, and that may have actually big consequences when patients will be receiving immune checkpoint therapy. Because again, those drugs are really unleashing everything. Whatever we are starting with, we will have more of it. So those small differences we have prior diagnosis or baseline will be actually amplified actually as patients are receiving basically immunotherapeutic drug, and that may explain why some patients are not doing as well and some other when it comes to actually clinical outcome. The second part we're actually working on as well, remember these differences when 1/3 of our patients were technically able to receive immune checkpoint inhibitor therapy, and only 21% of our African American patients didn't actually receive immunotherapy. How did we lose 10% actually of our patients at LCI? And to answer this question, we are going back to tissue this time that we have collected in 2016 from those patients. And we have actually developed like a condensed version of our IMP panel that is really designed for blood and single cell actually from the tumor, but we can also do some of it on tissue using pathology assay, histology. That's typically what we do when we want to look at a protein or a cell actually in a tissue. We'll be using actually a slice of the tissue from a biopsy or an explanted tumor, and we use actually color-coded antibodies that will determine if it is there or not. When the tissue lights up green, red, or any color you want, it will tell us if the protein or the cell you're looking at is actually there. So we have developed this IMP panel and validated it for tissue analysis. And we have actually also a biobank at LCI. We've collected over 15,000 clinical specimens, tumor biopsy from lung cancer patients between 2014 and 2016. We have about 3,700 of those. And from there, we are looking at clinical annotation, ensure that diagnosis is one cancer and one cancer only, making sure we know what drug patients have received, we know exactly clinical outcome, previous line of treatment, later line of treatment, to try to actually understand among 200 patients within this entire cohort whether we see actually biological differences between African American and Caucasian patients that would explain why, when 1/3 of our patients were PD-L1 positive, only 1/5 of our African American patients actually were ultimately receiving with a drug that we know is the best way to go forward. So that's all what I have. If you have any question, I'm happy to answer anything you may have. And that's geographic distribution of LCI center. We are all across.